First of all, welcome to the second GHGA webinar. We're very excited to have you here. We're going to be talking about research consent today with Andreas and Simon. Just as a reminder, we are recording this meeting so we can make it available on demand later. So keep that in mind if you want to speak up during the Q&A that you might get recorded. Since we're not currently organizing these webinars on a regular schedule, we would like to invite you to join our newsletter or follow us on social media so you will be informed about the next webinar happening. So here on the screen, I have a QR code that will lead you to the website where you can sign up to our newsletter. So since some of you might be new here, I just wanted to briefly introduce GHGA. It's the German Human Genome-Phenome Archive, and it's still very much in development. But something we're aiming for is GDPR-compliant human omics data sharing. And we're aiming for this to allow secondary use of clinical omics data in research, which could help with biological discovery and replication of findings. And this could lead to translation of new research insights and delivering more value in genomic medicine. And we're expecting this project to involve a lot of interaction between very diverse communities, like biologists, geneticists, scientists, physicians, patients, etc. And that's one of the reasons we're putting on these seminars, is to kind of reach all of these different communities and have everyone talk about these things together. So our speakers today, one of them is Andreas. He has a background in philosophy and German studies, where he got a BA from the University of Münster. He then got a master's in philosophy, also in University of Münster, and a PhD in philosophy and ethics, and is now a postdoctoral researcher at the National Center for Tumor Diseases here in Heidelberg. And he has been a member of GHGA since May 2021, where he is working in the ELSI workstream, which is the Ethical, Legal, and Social Implications Research workstream. Our other speaker today is Simon Parker. He has a BA in history and a master's in criminology, as well as a PhD in criminology. And he has been a data steward for GHGA since 2021 as well. And he is currently our data protection coordinator. Both of them are going to talk to you today about research consent and how you can apply it. And the way this is going to work is Andreas is going to give a short talk. Then we have 10 minutes for a Q&A session, and then Simon is going to launch into his second talk, and we'll have another Q&A session. And with that, I will stop sharing my slides and let Andreas take over.
Thank you. Let me share my screen. Okay, I hope you can all see this and hear me well. If there are any issues, please let me know. Yeah, Julia and Nicole, thank you first for organizing this webinar, and thanks everyone else for coming today. I'm going to take over the first part of this webinar, which is kind of a general introduction into informed consent in research. And it's always kind of challenging to make these kinds of talks because you don't really know how to how basic to keep the content. I hope it's not going to be too boring, and there are some interesting things for everyone to find. So I want to start off with a very familiar case, a familiar example, and that's the case of Henrietta Lacks, who is or was the donor behind the famous HeLa cell line. And just for a short recap of the case, Henrietta Lacks attended Johns Hopkins Hospital in Baltimore in 1951, and she had a biopsy there and was diagnosed with cervical cancer. She received then radium treatments, which was at the time the standard treatment method for cervical cancer. But sadly, she died later the same year. Before she died, though, some of her cancer cells were given to George Otto Gray's lab at Johns Hopkins without Lacks' knowledge or consent. And back then, Gray collected cells from all cervical cancer patients at Johns Hopkins. And it was the common practice at his lab to label the cell line using the initials of the name of the donor. So that's how HeLa came to be the name. And Gray found that Lex cells were quite special or unique in the way that they reproduced. He managed to successfully clone them, so the HeLa cell line became the very first immortalized human cell line. And from there, from that point onwards, the HeLa cells were distributed globally, were commercialized and used for medical research broadly. And they came to play a significant role in many innovations in the medical sciences. For example, the development of the polio vaccine and advances in gene mapping also played a role in the development of the COVID vaccine. And only in the 70s, the case became controversial because Leck's family, who didn't know anything about the broad use of HeLa cells in research, they began to ask questions after they received phone calls from researchers who were requesting additional blood samples from family members. And from that point, the controversy continued. It continues until today. I don't know if you heard about this, but last year, Leck's family filed a lawsuit. They're accusing, I think, upwards of 100 pharmaceutical companies of what's called unjust enrichment, which essentially means when you're trying to benefit off wrongful behavior or wrongful conduct. And it's really interesting when you read interviews with the family members, they often speak about a contrast between, on the one hand, being proud that their mother's or grandmother's cells had such a significant impact on medical science and helped so many people. I don't know if you can see that in the picture of the gravestone, but they even had HeLa written on her gravestone, kind of giving expression to that pride. But on the other hand, a sense of injustice about what happened and how the cells were used without consent so broadly in research. And I think that really nicely points to a general two sides of a coin situation that we have when we talk about research involving human subjects. It's always, on the one hand, that medical research has a huge positive social impact in making our lives as a whole significantly better. Each of us has profited from medical research. If you think about the fact that many of us wouldn't even be here if it wasn't for the advances in research and for the vaccines developed, for the dying out of certain kinds of diseases. And on the other hand, medical research just has the potential to raise ethical issues or questions or worries. And these issues are very diverse. So research has the potential to infringe upon basic personal rights, for example, autonomy and privacy rights. It has the potential to harm individuals or groups. A keyword here is genetic discrimination. That essentially means, for example, when genetic knowledge about you is leaked to potential employers or insurance companies, and then you end up not having access to certain kinds of jobs or insurance policies because of that genetic knowledge. And there are also more broader picture worries, for example, about potential exploitation of specific groups or communities, or in general, the unjust distribution of burdens and benefits of research. And the interesting thing about Henrietta Lack's case, I think, is that it really speaks of all of these or many of these issues. So, for example, there's the issue that Henrietta Lack had not been asked for consent to the use of her cells in research. There's the issue of privacy when we think about the connection of her name and the name of the cell line and the phone calls received by her family members. And an important part of the story is also, of course, the issue of exploitation. So Henrietta Lack is a Black woman in the 50s who attends the only hospital in her area that is treating Black people at the time, who then in turn often become research subjects unknowingly. There's some background noise now. I hope you can still hear me well, because someone decided to work outside. Yeah. Well, and yeah, so it's just the case back then that the Black community or Black communities are carrying the burdens of research, but on the other hand, the benefits of research don't really come around and come back to those communities. They have nowhere near the level of access to healthcare than white people have. So the issue of exploitation is very present too in this case. And the lesson from this conflict is, of course, not that we should stop doing research or so. The lesson is just that research requires proper ethical and legal guidance, regulation, or governance, with the aim that we can realize the positive potential of research and the benefits, while controlling the negative potentials to harm individuals or groups and keeping those at bay. And in this regard, consent can be just understood as an ethical governance measure. So consent just ensures that people aren't just used or treated as mere means to the ends of research, but that they are treated as participants who freely choose to contribute to the planned research activity. So basically, you could say that consent just turns research into a valid social contract. That said, I think that the Lex case also shows that consent isn't the whole of the story. So as I said, the potential ethical problems are quite diverse, and consent doesn't just solve them all. So imagine for a moment that Henrietta Lacks had given consent to the broad use of her samples in research, and this wouldn't make the other issues, for example, the exploitation issue, go away, right? It's just still unfair if certain groups carry the burdens of research without having access to its benefits. So the point here being that it's totally possible to give consent to research that then ends up being harmful or exploitative. And it's not the responsibility of the patient or the research participant to make sure that they only consent to research that doesn't have these features, because it's the responsibility that lies always with the research system. Consent is, of course, also a legal governance measure. And since we're talking about genomic research here, we are talking about the use of personal data, which is regulated under the GDPR. And as you probably know, if you are a data controller and want to process personal data and health data as well for research, then you need to demonstrate that you're doing this on a valid legal basis. And one way to demonstrate this is to demonstrate that you have the data subject's explicit consent to your data processing. So consent is one legal basis for data processing under the GDPR. And in any case, whether we talk about ethical or legal consent, that consent needs to meet a number of conditions in order to be valid. So I want to talk about these conditions for a moment. So you know that we often speak about informed consent and say that informed consent is required for research, but that's really just a way of highlighting one condition for valid consent, namely that it needs to be informed. At a minimum, it seems that consent needs to meet at least 3 more conditions. So first, consent needs to be capacitated, which means that the consent giver needs to have the basic capacity to make decisions for themselves. This is relevant, for example, in the context of conditions like dementia or so, where a patient can communicate decisions, but they might not be in a position to make those decisions. So it might be necessary to make decisions in their best interests for them. Then second, consent must also be unambiguous. That means the consent must be a clear expression of agreement to the indicated research action. And the third condition, consent must be voluntary. So it must be freely given without coercion or undue influence. And now to the informedness condition for valid consent, that's actually a twofold condition. So we normally handle that as one condition. Consent needs to be informed, but it's actually a twofold condition, because for a consent to be informed can mean two things. It can mean that I have been given certain information before I consent, and it can also mean that I have understood certain information before I give consent. So to see the difference between disclosure and information and understanding of information, you could think about like an everyday context in which we give consent. If you visit any website today, then the first thing you'll probably see is a pop-up window that asks you to consent to the use of cookies or personal data. And I mean, this is just a wild guess, of course, but I assume that most of us don't actually really read the terms before we press consent. And in this example, we have been given information and maybe also all the relevant information, but perhaps it's not fair to say that we have understood the relevant information, because we haven't even read it. And it's not even clear, I guess, how much the website host cares about whether I understood what I'm consenting to. Maybe it's more a means of protecting themselves legally that they give me that information. And in the medical research context, this should be, of course, or it is or should be very different. If the principle of informed consent is to have any value in research, then the focus must be on the understanding part of the condition. So it must really be that we should think about how we can give patients and research participants information in a way that helps them to gain a better understanding of what they are consenting to. And that brings me to talk about the challenges around research consent. So as you also might know, the default case of informed consent is specific consent. That basically means that I give consent to an individual well-defined purpose. For example, I'm not feeling great, so I go to see a doctor and she says, I would like to take some blood from you and look for this particular virus. Because the symptoms you have suggest that you might have this virus. And in this case, it's very easy for the doctor to tell me what it is that she intends to do, why it's important that we do this. And it's, on the other hand, very easy for me to understand whether I want this or not. And in the clinical setting, usually it will be quite obvious that it helps me. So it will be quite obvious that I want it. And this is an example from the clinical setting, and you might get specific consent in research settings too. For example, where the participant is consenting to a very specific research study or trial or so, where data is just used for one specific purpose. But in general, specific consent isn't really suitable for research today. And the reason for that is that in research, it's important also, as you know, that data can be made available to a broad research community, because in general, the more of these data resources are available to researchers, the more precise their analyses will be, and the more informative the research results will be. So what we need in research is consent to the long-term storage and broad use of data. In other words, we need broad consent from participants. And broad consent means that I consent to a wide range of partly unspecifiable future research purposes. And the problem, as you can probably tell, is how can my broad consent really be an informed consent? How can I have a sufficient understanding of what it is that I'm consenting to if I'm consenting to so many different applications of my data at once? And some of these applications can't even be specified because they lie in the future. that's kind of the major challenge around informed consent, informed research consent. And many ethicists still think that broad consent is sufficiently good, sufficiently good practice. Some deny that and propose other approaches to informed consent in research, for example, dynamic consent. Legally, broad consent is also tricky because the GDPR technically requires consent to be specific. That was on the last slide. I should have put it on this slide, I guess. I won't go into more detail there, but it's tricky to make sense of broad consent under the GDPR. Instead, I wanted to talk about the challenges more specifically around broad research consent. And one is, of course, that when we talk about genome data, many say that we are talking necessarily about data that is particularly sensitive and personal, because genome data can't fully be anonymized. And against this background, it seems that data subjects' consent is always necessarily playing a central role in genome research. So we can't really go away from the principle of consent. But then one practical challenge here is how to write comprehensible consent forms. So consent forms that really help people gain a better understanding of what they are in for when they consent to research broadly. And here I think there's really like a lot of room for improvement, especially in Germany. I'm now just talking from personal experiences, but my well, many German consent forms that I've seen are still rather lengthy and they're quite convoluted. They don't really use simple language and so on. So I think there's some practices that could be used more to achieve more comprehensible consent forms. Just to mention one example, one thing that I find that can really clean up or clear up a consent form that is convoluted is to think about the hierarchy of information, like what needs to be included directly in the consent form, because it's immediately relevant to the consent decision, for example, the risks to the data subject. And then there might be information that we can just refer to, and just say, if you're interested in, genomic research, here are some resources or so, just kind of outsourcing the information to other places if that's possible, that kind of stuff. Or for example, also using reference terms. There are lots of, I often see consent forms that use reference terms like we or us, and it's really not clear who that refers to. Like you would think it refers to the people who give the consent form to you, but then it sometimes just seems to refer also to external researchers, so like the whole research community. So really like having reference terms clear. So this kind of stuff. And another challenge is, I think, the lack of global standards. For example, when it comes to consent processes and consent requirements, at the moment, there's a lot going on internationally. Initiatives and projects are building infrastructures for global data sharing. And these global networks, of course, require us that we also think about harmonizing consent forms and the consent processes globally. So we really need to get away from like the local standards to some more global standards. And the last point that I wanted to mention about this is that we also need to think more about how to connect the regulatory level, so the legal and ethical dimensions of consent, to the technical level, because whatever we come up with solutions on the regulatory level will always be or will always have to be executed on the technical level. So for example, there need to be technologies to tag data sets and so on with consent information, but still make sure that public metadata is anonymized or non-identifiable and so on. So all these things connect. So the challenge is really to connect the ethical, legal, and technical dimensions. So this is just to mention a couple of challenges. And I'm coming to my last slide now. I don't know how I'm doing it time-wise, but okay, cool. yeah, just on the last slide, I really just wanted to point to some initiatives who are doing work when it comes to these challenges, and refer to some resources. So Simon is going to talk about our consent tools in a minute, but of course GHGA is a big consortium, and we work on different solutions from the regulatory level to the technical level when it comes to genome data sharing. So just visit our website, subscribe to the newsletter, follow us on Twitter, whatever helps to kind of keep you in the loop about news from GHGA, new tools, helpful resources. And there's also a few resources that you can go to when you're writing consent forms, for example. There are a lot of consent form templates out there. Wherever you are, there might be templates at your institution, and there are also some more general consent templates. I find that the German ones are always kind of limited in some ways. So, for example, the one of the Arbeitskreis of ethics committees, working group of ethics committees, is only in German. The MII, so Medical Informatics Initiative, has a broad consent a template, but that's only usable basically, or only approved for the MII governance context, so you can't just use that in other ways. Still, I think those are helpful resources when you want to know where to start by writing a consent form. You can just have a look and find out where to start. And internationally, there's also a lot going on. So there's, for example, the Beyond 1 Million Genomes project. They work on recommendations for consent forms, for example, and other things. All of this is work in progress, so you also need to just come back to that and see if they have any news on it later. And last but not least, the Global Alliance for the Genomics and Health, who are doing really great LC work. In particular, they have published some documents, some consent documents under free access, where you can get like general and more specific consent clauses for different contexts, ranging from healthcare to research settings, which are really helpful and also really general, so that they can be applied to lots of different contexts and specified for the particular context. And yes, that's it from my side. Thank you for listening. And I'm looking forward to your questions.